Okay, so this lecture is going to be on Beyond Mendel's Laws, Part 2. So we're going to look at polygenic traits, epistasis, and then move on to some mitochondrial traits. So polygenic traits is the first one. They are influenced by expression of the genes you inherit, but also they're largely influenced by the environment and how you go about your everyday life. So examples of this would be height, skin color, body weight, fingerprint patterns, and behavioral traits. So for example, some place you live might be warmer, so your skin color might be different, and what you eat could largely control what your body weight is. So it's not always just the genes that you have in your body, but also what you do to influence those genes. Moving on with polygenic traits, so it makes it very hard to predict polygenic traits because you can't always determine what the environment is going to do. So it is additive, which means that the traits are not always, or the genes are not going to be identical to what you got from your parents. The frequency is usually a bell-shaped curve, so it could be kind of normal, but also it bells out because not everyone is going to be the same that you got from your genes, so it's going to show a lot of different traits that can come out of the environment and the genes that you inherit. For example, fingerprint patterns are polygenic. So we have here dermatoglyphics is the study of fingerprints. So the number of ridges on your fingerprints is largely determined by genes and prenatal contact. But the average total ridge count for a male is 145, and in female it's 126. So we're going to look at why this happens and see what happens in males and what happens in females and why it's a polygenic trait and why we're so different than each other. Another one is height. So the difference in height of students back in a couple years and today are a lot different. It's been improved because diet and health are overall better. So height and weight have been getting better over the years. Skin color. So melanin protects against certain DNA damage from the sun and UV radiation, and that increases melanin synthesis when you do go outside, so then that's why your skin gets darker and you get tanner. Humans have the same number of melanocytes per unit area of skin, so it's going to differ in number from each other. Continuing on with skin color, the definition of race is based largely on skin color. It's used on a social construct more than biological concept. Skin color is not a reliable indicator of ancestry at all because it just depends on where you live, the weather, so it can change very frequently depending on what you do in your environment. All right, moving on to epistasis. Okay, so this is when one gene controls the expression of a second gene. So an example of this would be Bombay phenotype. Okay, so this is in your blood. So for the antigens A and B, there's a protein that's an H protein. When the H gene is in there, it's epistatic to the I gene, which would be your O blood type. So if you have an HH genotype, you would have no H protein, and therefore, if someone had an O blood type and they needed to get an O type blood, and that person would have an H protein on their blood cells, they would not be able to get that O type because they don't have the H protein and the other one does. Okay, so the H gene is going to basically control the second gene and what they're able to get from their blood typing. Another one would be eye color. Okay, so the OCA2 gene is going to be controlled by the HERC2 gene. It controls the expression. So if you do have it, like down here, if you are this genotype, it looks like you'll be brown. Okay, but if you have this gene right here, it's going to automatically make it recessive and totally just wipe out the gene that you had before. So that's going to overrule, so that would be epistasis when it takes over the other gene. All right, genetic heterogeneity. Okay, so different genes can produce identical phenotypes. For example, hearing loss, there are 132 autosomal recessive forms. So it can happen in a lot of different ways. So, and then some genes may code enzymes that catalyze the same biochemical pathway, so it can produce the same enzymes or different enzymes that are part of the pathway as well. So you can see down here it produced different enzymes. Phenocopy or phenocopy, everyone says it different. All right, trait that appears inherited but is caused by the environment. So for examples, down here, exposure to teratogens. This causes the limb to inherit a developmental structure, and then infection of HIV virus. It can be passed down to mother and child, but it's not actually inherited. It's just passed down. So if you get it and then you pass it down to your child, it's not actually inherited. All right, these are some examples of all the ones that we've talked about that are not single gene ratios. So we talked about lethal alleles, we talked about multiple alleles. So this is a good chart to like keep track of which one is which, especially when you are doing your project. So these are good examples if you would want to choose some of these, and it tells you what inheritance type it is. Another one is mitochondrion. Okay, so mitochondria, which we talked about, is the organelle providing cellular energy for our bodies. It contains a small circular DNA inside of it called the mtDNA. It has 37 genes on it inside of it. It has different DNA in our cells than the nuclei of our cells have. There's no crossing over. It has a high mutation rate, which is why there are a lot of mutations in the mitochondria. Moving on. So this is an example of a mitochondrion pedigree chart. So mitochondrion genes are transmitted from the mother to all of her offspring. Think about oogenesis. So you have the mother up here. She is going to give it to all of her offspring. Okay, and then all the mothers down here are also going to give it to all of the offspring. You can see that it's not passed down to the ones that do not have the mothers affected. So whenever there's a mother, it's going to be passed down. Heteroplasmy, okay, is the condition where the basically the mitochondrial DNA sequence is not the same in all copies of the genome. So each mitochondria will have different alleles for the same gene. In each division, the mitochondria are distributed at random. Think about mitosis and meiosis into daughter cells. Okay, basically in meiosis, it's going to get passed down, and it's going to be able to be distributed at random. So scientists use DNA from mitochondria to basically look at the past. They use it in forensics a lot. Okay, so they use it to see who was the suspect of a crime, identify war dead, especially looking at historical records. And then it's usually likely to survive extended damage in cells. So they look at the mitochondria DNA because it's easier to have DNA from the mitochondria than the cell, which is probably might be damaged a little bit. All right, that's it for this lecture. So if you have any questions, please ask, and please make sure you filled in all the notes.